Hi, welcome back. We're going to talk about toxins today, and here's our lecture outline. So, we're going to talk about a couple of case studies of toxins in the environment and how they impact humans or other ecosystems and wildlife. So, we're going to talk about lead and DDT, which are two common examples that are talked about frequently. So, we're going to revisit some of these basic concepts. We're going to talk about biomagnification in ecosystems, and then we're going to talk about bioremediation. So it should be a fairly straightforward, quick lecture, and that is because you're going to be doing some reading on your own and learn more about bioremediation and toxins in the environment on your own. Okay, so what are some of the risks of having toxins in our environment? There's a lot of negative impacts that toxins can have, and they can behave as neurotoxins, for example, and disrupt your nervous system. Lead is a common example of that, so we'll talk about lead first. And we'll talk a little bit about why lead is so bad for development and why it's such a negative thing to have exposure to it when you are either in utero or an infant or a child. Carcinogens are chemicals that cause cancer, and tetratogens are chemicals that will disrupt actually the integrity of your DNA molecules. So every cell in your body has a nucleus. Most cells in your body have nuclei and with DNA in them. What is DNA? DNA gives your cells the instructions of which proteins to make and what processes to run. And if you go in there and start disrupting your DNA with things like UV radiation or smoke, for example, if you are a smoker, in some cases, when you are developing, so alcohol can affect you negatively if you're a developing fetus, and disruption of your DNA can have really bad implications. So one thing that can happen is the cells can become cancerous, for example. Okay, so let's talk about lead. What are some sources of lead in our environment? So a very common source of lead is through pipes. So the pipes that bring the water from an aquifer to your home are made out of certain metals and have many layers of things that they are coated with, and over time corrosion of the interior of the pipe can lead to the exposure of lead, and then the water is going to start carrying lead into the drinking system and into the faucets of people's homes. And there is some talk in the news currently about lead contamination. So one area that people have been discussing very frequently is Flint, Michigan. And they've been talking about high levels of lead in the water. And if you've been sort of paying attention, you might have heard that this is considered a pretty bad crisis. And it's probably extremely negative, and it's going to have a lot of negative impacts that people are being exposed to this contaminated toxic water. Okay, so that is lead in your drinking water. What are the exact levels of lead in the water in Flint, Michigan? So this graph here is comparing the levels of lead in the water to other Michigan cities, and they are showing that after a certain point, the lead levels in Flint, Michigan actually did rise above the lead levels in the water in other areas. So there is a level that the government recommends not to go beyond, and I think by all accounts, these levels at times have exceeded them. So what is so negative about lead? During development, so lead is going to affect a developing child a lot differently than an adult. So adults can experience toxicity, but a child will actually take in the lead, and it will impair proper development of the brain. In my opinion, that's probably the most important effect it can have. But it can also impair the proper development of a lot of other organs, and improper development and lead interfering with the development of these elements can have the outcomes that are described in this slide here. So, for example, one very strong link of lead exposure to children has been linking that exposure to lower IQ levels. So actually having lead impair the development of a brain properly is actually going to lead to intellectual disability in a lot of cases. ADHD, hearing loss, damaged nerves. So I'm just looking at the brain section over there, and you can look over the other effects that lead have on development on your own. And we are proper scientists. We don't just go on the words of nice infographics like the one I just showed you previously. Even if it is cited from a relatively reputable journal, we like to see actual data. And this is, I think, an important concept for you to think about when you think about your own health and the effects of your own environment on your own health. So let's say you are curious about some medical process that your doctor recommends, or some medical process that your friend recommends who isn't a medical doctor. It's important for you to be comfortable reading scientific literature. And I think that the practice that we've had over the last few lessons, where you have been approaching the literature, and the practice that you'll have today or this week with the literature that I've supplied this week, is all for you to learn to be comfortable reading scientific literature, and also not taking everything as you hear it, without being able to also research the actual data that supported people's conclusions. So that's a really empowering and important element for you to have as just a important as a human being, as a active person who makes decisions about themselves in a informed manner. Excuse me. Okay, so here are the papers that actually look at these the link between exposure to lead and different behavioral or intellectual problems that resulted as a result of that exposure. And these are pretty big studies, so they're looking at tens of thousands of children at once. When you have this kind of after the fact ability to look at different areas that what the concentration of lead is in the drinking water, and you can look at very large groups of people after the fact, that's a really powerful technique to run a clinical investigation that way. Of course, if we are interested in the effect of something toxic on human beings, it's a lot better not to have the exposure, right? But because this has already happened, it gives us, statistically speaking and scientifically speaking, very strong ability to find links between this particular toxin and the effects it had on people. Otherwise, what you'd have to do is start giving people toxic chemicals in a laboratory setting with controls, but usually these kind of studies are not approved for reasons that I'm sure you can think of yourself. And what are the levels of lead that have these effects? So this is another nice cartoon. Of course, I'm telling you, don't trust cartoons, so you can always go back and look at the source, and also try to find some literature with actual numbers where people have looked at the actual numbers. So 10 micrograms per deciliter down here are the levels where we start seeing of neurological symptoms, of resulting in neurological symptoms. So keep that number in mind, 10 micrograms per deciliter. And we can see with higher quantities, at some point, you're going to have extremely severe reactions. So you can have acute lead toxicity, but in general, we're talking about lower level exposure rates. So that number in mind, 10 micrograms per deciliter. So if we look at this side of the graph here, 10 is about here, right? So we can draw a line this way, and we can see that actually, look at that blue line there, that's the amount of lead that was in the water. And this is a nice timeline of the history of lead, and this kind of awareness, this timeline reoccurs over and over again when we study toxins in our environment. So we have some discovery that humans discover a new chemical or a new process that is wonderful and very efficient, and they utilize it at very high levels, and all of a sudden it becomes very widespread without the realization that they're actually adding a fair amount of toxins and pollutants and dangerous chemicals into the environment. Then, once people realize that this is actually having a bad effect on the local environment and their own health, they start putting legislation in place to regulate the abundance of these chemicals and the amount of chemicals that are put into their environment. And once we implement these regulations, there is some turnaround time, so it takes some time for people to use up the chemicals that they already have, and for the manufacturing to stop, and for other replacements to be used, right? For all of these pipes, for example, to be replaced, etc. And after that lag time, we do see actually that the toxins in the environment go down. However, of course, there are always long-lasting effects, even once the legislation has been put into effect. So I think this really hammers home the point of how important it is to take action early on, because the ramifications will always last longer than we think. In the 70s, people start stopping the amount of lead being produced, the amount of lead being put into gasoline. They stop putting lead into residential paint. Now, this doesn't mean that that there is no paint in any lead-containing paint in any residences. Of course, a lot of residences still have paint that contains lead in it, which is considered pretty hazardous, especially if there are children around. So they start banning all these these sources of lead in the environment, and all of a sudden the amount of lead that is found in people's blood starts going down. And it's actually quite shocking if you look at how much lead was in people's blood before these preventative measures were put into place. So we're around here, and think about, well, I was born here-ish, so, potentially anyone my age or older could have had a pretty significant exposure to lead. And here's just a similar picture showing you the amount of lead that is detected in blood levels of children now. So here's the graph of the higher level of lead in the blood, so 10 micrograms per deciliter, and they're saying that this is the percent of children that have this level of lead, and over time it has gone down, so that's very optimistic. And this is the 5 micrograms per deciliter, and that has gone down as well. And you can go back to this source and read the article if you're interested. Okay, so that was an important study of a neurotoxin, a chemical that disrupts the nervous system and disrupts the proper development of the nervous system. Let's talk about another dangerous chemical that is actually dangerous in a lot of different ways. And that chemical, I'm sure you've heard of it before, and I'm sure it's come up in other environmental classes that you've taken, or other, anything that you've read about this subject, which is DDT. So this DDT follows a very similar timeline of lead, in that the widespread use of it was promoted very heavily early on, and then once people realized how toxic it had the potential to be, there were rules put in place, and there was a lot of implementation of new laws about using DDT, and then slowly over time the use was made obsolete. But there are still effects that have impacted the environment long term. So here's a fun little magazine article telling you how amazing DDT is. DDT is good for me, and the cows are singing, and the potatoes are singing, and everyone's happy. And if you look up pictures of DDT use from this time period, you see people just spraying it on all over human beings because of it was very effective against insects, right, lice and things like that. And they were spraying it all over their chemicals, and they were spraying it all over their home. So it was used basically to prevent pests and kind of bug. And you might think, well, something that is that toxic and is just killing all of these different different living things on impact can't be very good for anything. But people just did not have that awareness, and we now know a lot more than we did then. So, of course, this fun little cartoon is a bit bittersweet in retrospect. So, one of the initial reasons people decided that DDT was very negative and worth banning was because it was affecting what is somewhat of an American symbol, the bald eagle. And this is just a hilarious picture that I love of bald eagles. I spent a year living in Virginia, and there was several there. I lived kind of in a rural area, and they're really not very graceful. They would sit on branches and trees, and they'd fall out of trees, and you would just see a clumsy fat bald eagle like falling down a lot of branches in a tree. It was quite inelegant, to say the least. But since they are considered to be a symbol of, I'd say, American patrioticness, and they're present on a lot of our symbols in general, when we discovered that DDT was really negatively affecting them, people took action pretty rapidly. So it just goes to show you that for the right reasons, people will care, as long as it's something cute and important, right? I'm just kidding. Okay, so here is the quantification of bald eagle. I think these are mating pairs that they're counting, although I have seen different numbers on different graphs. But let's, for now, consider the y-axis here to be mating pairs. For example, it could be a different number, but if you just forget about the raw numbers, we're just looking at, obviously, some part of the population that over time was quite low, not only due to DDT, also due to other human activities like hunting and other effects that we had on the environment. And however, once DDT was implemented in a very widespread way, there was a lot of impacts on the bald eagles that people started observing. Most namely, their eggs became very brittle, and they would break before they hatched. So, and that was something people could see visually very easily. So people did notice the impact on bald eagles very early on, and there was one other very critical element to changing the way people distributed this very toxic chemical, which was a book that was published by Rachel Carson in 1962, and she, in that book, actually describes a lot of the effects that we were having on birds. So she also kind of focused on bald eagles and bird and other animals similar to bald eagles. So their number, when DDT becomes widespread, bald eagles numbers go even lower than they were previously, and EPA starts implementing rules about DDT use and how it can be distributed. DDT is classified as a probable human carcinogen, and we stop using DDT in large amounts, and we can see that now the bald eagles are thriving and increasing in numbers, and are free to, and have really been taken off the endangered list. So it's a bit of a success story. Here's my question to you. When you see this kind of growth rate, right, that looks kind of like a J shape, what is that growth rate called? It's very characteristic of an animal that is being recovered from almost from near extinction. So here's Rachel Carson's book, and there's Rachel Carson. And it's important to keep in mind that one person can have a pretty pivotal effect on a lot of different things, right? If you think about the person who wrote this book and had a extremely positive impact on the environment, or the person who lobbied our Congress about making sure that people were not aware of how dangerous cigarettes were, right? One person can have a big effect on a lot of different communities. Let's talk about biological magnification. So when you release, or not you personally, but when a person or a company or an entity releases toxic chemicals into nature, it might be distributed with a pretty low concentration, and you might be thinking, well, I can dust my plants, it's not that bad. But what happens is that over time these toxins accumulate in animals as we go up and up the food chain. And this is a concept that you've already been exposed to in your Symbio module. So this is something I expect you to already have a pretty good grasp of. Let's just remind ourselves why biological magnification happens. So do you recall this highly inefficient energy transfer between trophic levels of the food pyramid, as it were? If you look at the amount of energy in primary producers and how much of that gets transferred to primary consumers, the amount of energy that the primary consumers are eating compared to the amount of mass that primary consumers actually put on is quite small, right? So if that zebra is going around eating hundreds and hundreds of pounds of grass, it's only going to put on a pound or two of meat or of weight. So all of that DDT or whatever toxic chemical we're talking about is slowly being accumulated in that zebra over time. So that's bioaccumulation. But when that zebra has a certain amount of DDT in it, and let's say a lion comes along and eats that zebra, that toxin, whatever toxin is being biomagnified, is going to be even more highly represented in the lion, because now the lion has to eat a lot of zebras just to put on that pound of mass. However, all of that zebra is going to have a lot of concentrated toxins in it, which are going to be even more concentrated in the lion. There are a couple of cartoons about biomagnification just to give you an idea of the numbers that we're talking about. And again, this was in your Symbio module, so this should be hopefully already familiar. So if we're looking at parts per million of PCBs, we can see that in phytoplankton, the PCB concentration is about 0.025, and then zooplankton, which eat the phytoplankton, it's about 5 times higher, and then in the small fish, it's about 10 times higher than that, and then that's 5 times higher over that, and then once we reach the top of the food chain, it's now almost 25 times higher or more. So now we've gone from 0.025 parts per million to 124 parts per million. So that is a really big increase. Now, this is why when we consider the things that we eat, actually, it'd be a lot healthier in terms of toxins accumulated in your food to eat things that are lower on the food chain. So that's why they say not to eat tuna in very large amounts, or not to eat tuna very frequently, because the tuna is going to have mercury levels on the order of things that are at the top of the food chain here, whereas if you eat sardines, they have mercury levels that are quite a bit lower. Hopefully you are aware now that biomagnification is the increase in a chemical concentration in animal tissues as that chemical moves up the food chain. Now I just want to show one more because I found so many of these nice cartoons and I couldn't decide which the best one was. What I like about this one is that it's linked to a paper, and if you're interested, you can go back and find this paper. So they're actually showing you. So, we said before, we're not going to go based on cartoons. We want to see the actual data. And I know that you are intelligent and capable of making decisions intelligently, and you're not going to take somebody's word for something unless you see the data. And now that you are also a seasoned reader of scientific articles, you are able to interpret these articles, at least the important points, and take your own conclusions from them. So that's what I like about this paper. So we did mention that these toxins persist in the environment for potentially long periods of time after humans stop introducing them into the environment. And that is because they have very long half-lives, like a lot of the other things that we talk about, like plastics, like ozone, right? How long do volatile organic compounds survive in the ozone layer? Do you recall that number? I believe that'll be information for the exam coming up. So if we look at DDT can exist up to 30 years in the soil. So that is a very long half-life, and that's why we want to be very careful with the toxins that we introduce into the environment. Bioaccumulation, as we mentioned, is just the increased concentration of a chemical over time in an organism. That is the basics of the concepts that you are going to need for the reading. We can talk a little bit about scientific trials, so let's just go through a couple of concepts very quickly. We can study how chemicals affect organisms, and usually scientists are not going to take groups of human beings and expose them to toxic chemicals on purpose with the intention of seeing how those toxic chemicals affect human health. That would be considered extremely unethical, and I'm not saying that it's never happened, but it'd be very difficult to get that study approved in the current state of science in the United States. So how would a scientist study the effects of a toxin on humans? Well, one thing they might do, well, how would you do it if you had to conduct a research study? One thing scientists do frequently is they use other proxies to see how the toxins affect them. So mammals are a good place to start since we are mammals, and a mammal that's really easy to use is a laboratory mouse. So sadly for the mice, we use them a lot and expose them to toxins to measure something like an LD50. So what is the lethal dose that kills at least half of the mice that are being exposed to that toxin? That'll tell you the relative toxicity, meaning if you have the LD50 of something like fentanyl, you can compare that to the LD50 of something like DDT. So how do those numbers compare to each other? Is that something that you can figure out by reading the literature? And the LD50 is going to vary widely. So I can tell you for a fact that the LD50 of fentanyl is going to be really, really low, because I know that it takes very, very low amounts of that to kill somebody. So when you hear people in the news talking about pounds of fentanyl being captured or being found on, in some situation, and how that's enough to kill 40 million people or something like that, that's because the LD50, the lethal dose, is really low. We can also consider the ED50. So if the toxic is not something that necessarily leads to death, but we still want to know how toxic this compound is, we might talk about the ED50, so the effective dose that causes 50% of the animals to display harmful but not lethal effects. And why are we using this 50 measurement instead of 100, for example? It's because you get this asymptotic graph, and the 50 just is a nice convenient way of it's very easy to find the 50% point on this graph, whereas finding the 100% point is difficult because this kind of graph might approach 100 but never actually hit it. So that's just a theory behind that. Okay, so different kinds of studies we can do. We can do acute studies for the short term. We can do chronic studies to look at long-term exposure to a chemical. Sometimes that's more of a low dose. And importantly, Epidemiology is the field that studies the causes and patterns of disease in a population. So, if you're interested in the effect of lead on the population and you don't want to give people lead in a laboratory setting, but unfortunately, or fortunately for you as a scientist, there has been some exposure of lead in a community, you can go back and do an epidemiological study and study how this might have affected this population after the fact. So that would be a retrospective study, which would be one that is looking back on things that already happened. So a retrospective study looks at a chemical and how its effects have been manifested in the past, and a prospective study monitors a population and that might become exposed to chemicals sometime in the future. So if you're interested in epidemiology, it's a very very interesting and very demanding job market, and you can look into that. I think it's very, very interesting, and I think it's a satisfying line of work as well. Okay, bioremediation. So we have all these toxins in our environment, and we don't want them to be there anymore, but it's not very easy to filter, the entire water supply of Flint, for example. So what if you have contamination in the origin of the water supply? What if you have chemicals that are leaching into groundwater because they are present in the soil? So one thing people do is they use bioremediation to detoxify the ecosystem, and this is the use of bacteria or plants or fungi or other organisms that can take up and sometimes metabolize the toxic molecules. Not always, sometimes they just take them up and then we can harvest the plants or the bacteria, we can filter the bacteria and filter that toxin out. This just demonstrates how important it is to have that biodiversity and to have these organisms available to us. So when we talk about biodiversity being important, this is one of the examples why biodiversity is so important. We need these organisms available to us to be able to maintain our ecosystem and also to address some of the issues that we've been introducing into the ecosystem. So biodiversity is obviously very important, and there might be bacteria on Earth that can, for example, metabolize plastics or metabolize oil and get rid of contaminants that we've introduced into the ecosystem that we haven't even discovered yet. So maintaining biodiversity is really important, even from a very selfish perspective. So here's a nice example of bioremediation, and you can go to the website. This is in the additional resources where the group from, I want to say Stanford, but I'm not 100% sure I'm recalling that correctly, is studying the bioremediation of uranium in uranium water or uranium contaminated water with bacteria. So what they do is they have these pits. So you have uranium in water, and it's uranium is radioactive, so you have radioactive waste in the water system, and you don't want that to get into the drinking water, and you obviously don't want that to be drinking water. So what do? How do you get rid of that uranium? It's very, very difficult to just filter it out. But this group has found bacteria that will take up that uranium and basically incorporate it into the bacteria's own molecules, so into their proteins or whatever they're incorporating uranium into. And then when the bacteria die, they settle to the bottom of the pit. Much like, I think this was another symbio exercise you had, where you could see that when those bacteria go down to, when they die, they settle to the bottom of the lake. And similarly, when we have phytoplankton, right? Phytoplankton are taking that carbon from the atmosphere, taking that carbon dioxide, and then when they die, they settle down to the bottom of the lake, taking that carbon with them. This also, the bacteria, when they settle to the bottom, are taking that uranium with them. And then once that uranium is on the bottom as a mass, it can be cleaned out because it's no longer just dissolved in the water in a way that we can't filter it out anymore. You can go and read all the studies that this group has done. Very wonderful, important studies. And one of the things they found was that for some reason, for reasons that you don't necessarily need to understand, but the group is playing around with adding different nutrients, and an important element of this that you should be able to be familiar with is a limiting nutrient. So they add nitrates and they add sulfur and they add all these different kinds of nutrients, and they do find, oddly enough, that ethanol is one compound that will actually increase the ability of these bacteria to take up the uranium. And the group plays around. They also show that if you add more nitrates, the same thing happens. But they realize that if you add more ethanol, the bacteria take up the uranium more, or they divide more, or for some reason there's less uranium in that water source after they add ethanol. So and they really go into the chemistry of it, and that's obviously not important for you to understand. But it is kind of wonderful and interesting that they have figured out a way to use just bacteria and some other chemistry to get all of that contaminated water and clean it up. And they show pictures on their website of contaminated water versus clean water, and it looks completely different, and also it's now safe to drink. So that's obviously the upside that we are all hoping for. And that is our Toxins lecture. Thank you so much for listening.